Hi, I'm Dr. Amy Turner, and welcome to Protein Text Podcast, the platform where we talk to life science leaders and get to grips with the big questions of the day that help us to read the book of life. Today, we're thrilled to be joined by someone who is fundamentally changing how science is done, and more importantly, how it is trusted. Dr. Anita Bandrowski is a neuroscientist, data infrastructure pioneer, and the driving force behind tools that promote transparency, reproducibility, and rigor in research. She is the founder of the Research Resource Identifier Initiative, or RRIDs, and the CEO of SciCrunch, where she's building platforms that help to make scientific resources findable, trackable, and more importantly, reliable. So from the neuroscience lab bench to transforming how major journals evaluate manuscripts, Anita's work is reshaping scientific publishing and practice. So if you've ever wondered how we can actually fix the reproducibility crisis or what AI tools can do for scientific integrity, then this episode is for you. So Anita, welcome to today's podcast.
Thank you so much for having me.
No problem.
Really appreciate it.
So I've just introduced you there, but maybe you can give us an idea of what your scientific background is and how you got to where you are now.
Absolutely. So I have studied learning and memory in graduate school, and then I studied epilepsy in postdoc. And both of those were what we call small science, right? We were looking at individual channels. We were trying to really atomize as much as possible the questions of what is learning and memory, what is epilepsy, take it down to its most root level. Mm-hmm. But I would contrast that with what I do now, which is trying to look not very deeply, but extraordinarily broadly across all of science. So I moved from the bench to this kind of big science, open science, high throughput science when the first genome came out. Right. So I worked for Craig Venter for a little while in a company called Celera, which way back when actually, was able to put together the entirety of the human genome a little bit faster than the big effort in academia. But one of the things that we had not understood at that time was what to do with that information. Mm-hmm. So there were lots of people who were trying to figure out what to do with that information. And one of the kinds of things that they knew that they needed was to attach as much information and metadata to that entire sequence as could be brought to bear. Mm-hmm. So that's where we really leaned on the project that was, the Human Genome Project, with the entirety of the academic enterprise of the world, assigning individual functions and locations to genes. Yeah. So that's what kind of led me into bigger science. But of course, when you go bigger, you can't go deeper.
Yeah.
Right. So then later on, I continued in that vein. I came to work at the University of California at San Diego when the Neuroscience Information Framework was funded.
Right.
And this was a big project. It was a collaborative project among 5 universities. We all had to learn how to do bigger collaborative science at that point, which was great. It was a steep learning curve, but it was a lot of fun.
Yeah.
And we had also a different mechanism than I think most scientists have to deal with. That mechanism was a contract as opposed to a grant. And that contract, that's very important because in a grant, you can say things and you might not get to do them.
Yeah.
But in in the contract mechanism, you absolutely have to do the things that you set out to do. So one of the things that we had set out to do, because it was in the contract, was there was a small line in the contract for the Neuroscience Information Framework that said, hey, scientists are wasting a lot of time dealing with trying to find antibodies in the scientific literature. Mm-hmm. You should create an AI tool that would help these scientists find that.
Okay.
And so we had undertaken that work in about 2009. Mm-hmm. And by about 2010, we had discovered that there is no way that we could do that. And so we had to go back to NIH, our funder, and say, hey, we really cannot do this. People are not putting enough information into the manuscripts so that we could actually find those antibodies. There was about a 10% rate of addition of the catalog number into the literature
10%.
At that time. Yes. When we looked at and I mean, this was well before, a lot of the open access journals. This was before a lot of people were able to text mine. The licenses weren't there yet. So we were doing all of this sort of in a dark age of
15,
of
16
AI.
years ago, though, doesn't seem that far away for people to only be putting catalog numbers in 10% of the time.
Yeah, no, that was very surprising to us as well.
Yeah.
And at that point, we basically said, okay, how do we do this? How do we even know how many antibodies there are out there for people to use? And that led us, so instead of text mining at that point, what we did is we brought together a database, which you now know is the antibody registry. And with the antibody registry, we had basically gathered all of the information that we could get our hands on about the antibodies that existed, commercial, non-commercial, antibodies from people's labs. We tried to make it as easy as possible for people to add information into that database. And so we at least had a sense of how many antibodies are out there for people to even use.
Yeah.
And at that point, we weren't really sure what that entire ecosystem even looked like. So after that, we said, okay, but what we want to know is which ones are being used in which paper.
Yeah.
Right? Because the whole idea is if you don't have enough information about the antibody, you can't possibly figure out which one is being used in a paper, even using AI, because you'd be guessing.
Yeah.
And we don't like to guess. We're scientists, right? At our core, we're scientists. Mm-hmm. So what we really wanted to do is change the culture around antibody validation, around antibody identification. Yeah. And with that, and with the deep frustration of the entire biological community at that time of not being able to find antibodies, we started the RRID initiative, which is basically getting enough journals. So at the time, we had 25 of our journal editor-in-chief colleagues, brought them together for a series of meetings. And those journal colleagues had basically enabled us to use their journals as a bit of a playground, I would say. And they were asking, so what we said is, please ask your authors to identify antibodies by using this number, this RRID number. It was the antibody ID that we had used. And they did so. So they did that in many different ways. When the first 100 papers came out, we looked at how the RRIDs were being used, by whom, what was the accuracy of these things. And we had figured out that it was with that first 100 papers that came out that the accuracy for actually getting the catalog numbers and getting the RRIDs in improved the ability to find those products from, that 10%. What we had published is that 50% were findable, basically. So, if somebody was to go in and search the catalog, they could basically track down half of them, even without the catalog number.
Amazing.
Which was amazing. But we moved from that to 90%.
Wow.
Which was a huge leap for those papers. And all we were doing is asking the author to do this. And I had then started to take this on. And actually, the fun part of this is I think we're a bit away from 2014. So what I like to say is that we are currently in the 11th year of a 3-month pilot project with these journals because, in fact, what happened is the journals only agreed to a 3-month pilot project.
What was the reservation there?
Well, they didn't want to, go all in on something that maybe wasn't going to stick around, but they said we would try it.
Okay.
We would try it for 3 months.
Yeah.
The fun part is none of them really ended that
Good.
trial, which was really good. And but none of them had said, yes, we're really on board at the same time either. So they just said, well, can we just continue?
Yeah.
And we said, sure, of course, no problem. And we're in the 11th year of a 3-month pilot project.
Pretty successful pilot project, I think.
I'd say it was a successful pilot project as well.
So this was, you mentioned 2009, you set it up. 2014,
we actually...
big paper came out about antibody validation.
Yes, there were a lot of... so in this period of time, there were a lot of questions about rigor of science, about what was happening. And really, a lot of the big journals were the leaders of a lot of this reproducibility crisis in science. They were the leaders unwittingly, in a sense. They had reduced the space for the methods section to the absolute minimum.
Okay.
And when they did that, what happened is they became bits of advertising about the real work as opposed to descriptions, deep descriptions, scientific descriptions of the real work. And one of the journal editors that is now actually working for Chan Zuckerberg Initiative, but she really changed the way that we were able to describe science, because what she did at Cell Press Journals was she said, look, we are not going to put the methods section in this giant methods that you would like. But what we are going to do is we're going to make sure that you put in a reagent table into the front of each method section. And then what we're going to do is we're going to take the entire method section, put it in the supplement, but that supplement can be as long as you want.
Perfect.
And so what she was able to do, this is Katja Brose, and what she was able to do is take one of the big publishers and basically say, you will have the space that you need to describe this experiment in just as much detail as you possibly can. And when she did this, the other publishers started to come along and do similar things. They didn't all do the same thing immediately, but it was a really good example for what could be done with one of the big publishers.
It makes sense. It's like if a journal restricts the method section, in my mind, that's like having a recipe book and saying, hey, you can make this meal, but I'm not going to tell you what ingredients are in the meal. But this person's come along and gone, actually, it's pretty fundamental that you need to know the ingredients to go into a meal before somebody can reproduce that meal.
Exactly.
Yeah.
It's the same sort of thing. So that's why I say it was kind of unwitting because they had page limits on these articles, and a lot of these articles were, they shrunk down. So each of the figures was like 12 panels, with 14 insets. So you couldn't possibly actually describe that full amount of work in that 2-page document, which they all kind of required. So what ended up happening when we were, when I was a postdoc and when I was a grad student, if you wanted to publish in one of those big journals, you would almost have to publish a companion paper in another journal where you got a lot more space to actually write out what you did. But that sort of drove science in a really bad direction.
Yeah.
And so Katja Brose and others in that room were able to sort of pull that back, which was great because that has started to fix. I mean, you don't write a ship like science in 5 minutes.
No.
But you can actually start to pull. And if you pull in the right direction, people will come along, and they have.
Yeah.
So this has been an incredible journey for me because I've seen this writing of this ship. It's not quite there yet, and we won't be there because science is never perfect. But as long as many people care about that science and about what it is that they're doing, they care about the quality of what they're pulling out, I think we can right the ship.
Yeah. I suppose at this point as well, was this the point 2014, 2015, 2016? I know there was the big flagship paper about the reproducibility crisis. Maybe
Yes.
you can kind of talk about what that looked like at that time and, what was the reproducibility crisis? What is the reproducibility crisis?
Well, so with antibodies specifically, the reproducibility crisis was multifold, right? One of the biggest issues I had already talked about, we couldn't find which antibody was being used.
Mm-hmm.
If you don't have that piece, you don't have the recipe. And so you can try to reproduce something, but if you don't know the reagents, it can't be reproduced.
Yeah.
And in fact, I had talked to many scientists who, took me to the side and said, I did get a PhD and I think I got the right antibody, but I wasn't really sure. I've always been concerned about it. And if somebody goes back and sees that it wasn't the right antibody, then I'm not sure what to do. Other people had not been able to reproduce big papers and they had dropped out of science and they actually went into roles at antibody companies and other places.
That is wild. I think I read a stat recently that was, I think it was Amgen maybe tried to reproduce 53 major cancer hallmark studies and could only reproduce 11% or something like that.
Yes, it was 4 of them. Exactly.
That's wild.
Yeah. That was Glenn Bagley's work. And basically at that point, we were saying, look, we can't reproduce these big papers. Mm-hmm. So again, if you cannot reproduce the recipe, you cannot reproduce the results because the recipe didn't contain enough information. Yeah. And the cancer reproducibility piece was one of the big reasons why we got to know about the reproducibility crisis. But it also gave us a method to be able to see the reproducibility crisis and see what are the ways that we might start to address how we can fix the problem.
Yeah.
And Glenn Bagley pointed us to a set of practices that should help. Mm-hmm. And he basically called everybody to do better. And I think people did heed his call. If you look at the second version of that same reproducibility crisis in cancer paper, which is so the problem with the Glenn Bagley work is he was working for Amgen, and Amgen did not release all of the data that Glenn Bagley was writing about. Mm-hmm. So he couldn't release that because it was private to the company. But there was a big reproducibility effort in cancer that was fully open and fully transparent. And that was led out of the Center for Open Science. Right. So that effort came several years later. It was just published now a couple of years ago with all of the papers. Everything was pre-registered. One of the interesting things about that is when they went to reproduce that work, 0% of the papers that they tried to reproduce, that recipe, 0% were able to be reproduced without contacting the author.
Oh my gosh.
So that was one of the kind of eye-opening shockers.
Yeah.
Because yes, some of those papers were reproducible, some of them were not reproducible, but the fact that you couldn't start without the author's help, it sort of begs the question.
A lot of innovation.
Why are we even publishing these things?
Yeah.
If we can't, actually write out our recipe. Mm. So that start is where, and I mean, science is hard. Science is hard. And just because I can't make a filet mignon to save my life doesn't mean that filet mignon cannot be created, right, by someone else who's far more competent than myself. But the fact that I can't make the filet mignon doesn't mean that it's not reproducible. Yeah. It's not reproducible in my hands. And that's for sure. I've tried. And I will say, I am not good enough for this. But it can be reproduced as long as you have the right training.
It's interesting, actually, that you said I am not good enough there. Just going back to what you said about people not being able to reproduce something and then leaving science. Yeah. That's something that I'd never thought of before. Like, imposter syndrome is massive anyway in science. And especially if you're really trying to reproduce something that's written down, somebody who you highly respect in the literature is doing it. You're thinking, I want to meet this person, I want to be in their network, and you can't do it. Obviously, that's going to have an impact. So it's interesting that even, psychologically going back to RRIDs, having something that's going to just make that step a little bit easier is fundamental, really, for driving innovation.
Exactly. It's fundamental for driving innovation. It's fundamental in being able to have science that builds on itself from paper to paper, from lab to lab, as opposed to just building within your own lab because that's all you trust.
Yeah.
Right. So we want to be able to trust our colleagues, but how do we make sure that the papers that we're putting out, the reports that we're putting out about the data that we're gathering, how do we make sure that that is a trusted piece of evidence that I have done something that is substantial? I mean, again, just because I can't make the filet mignon doesn't mean that it's not a reproducible protocol.
Yeah.
It can be a reproducible protocol. I'm just not good enough at doing it. But if I at least have the recipe, all right, it might look something like what we've seen with the failures of cake making, but at least there's enough there so that you can actually figure that out.
Yeah.
And a much more competent person would be able to do such a thing.
So you mentioned, trusting the publications. You were also very instrumental in forging the 5 pillars of antibody validation so that we can actually trust reagents as well. Could you maybe touch on what these pillars are?
So there was a really wonderful working group way back in 2015, 2016, which tried to figure out how do we even look at the concept of validating an antibody project, an antibody experiment. And by we, I was part of the group. I was in the room. I was very pleased to be in the room. I was happy to be there. My little voice was, hey, you also need to make sure that you write down which thing you're using. Yeah. But there were lots of great people there who knew antibodies very, very well. They ran large projects. They ran companies. They ran smaller projects. They made antibodies on a regular basis, and they knew how to validate. And we essentially took a set of surveys with hands up and what have you around the room. Can you validate an experiment for Western blot like this? There were hands raised. We talked about the various aspects of that as needed. So we took sort of the temperature of the people who really knew antibody experiments. We figured out what was the consensus. And the 5 pillars really emerged from that consensus of the experts in antibodies, what was able to be the gold standard. And so the most important piece of the validation pillar paper was a gold standard emerged. And that gold standard was if you have a knockout cell line, for example, or animal, you can take that gene out. And if that gene does not exist in your sample, then it also should not be bound by any antibody.
Yeah.
So that became the gold standard for validation. But there are other pillars. You could use multiple pillars. One of the pillars that is possibly one of the easier but a little bit more problematic was the pillar of using 2 different antibodies. Mm-hmm. So in this case, identification is very important because even though you might think you have 2 independent reagents, sometimes you actually have the same reagent in your hands. And so it's not able to be validated against another.
I think this is a really important point to raise, actually. I just want to kind of give it a little bit of airtime because I know when I was in the lab, if an antibody didn't work in my hands, obviously, again, imposter syndrome. It's me. It couldn't possibly be the reagent. But then somebody goes, maybe it is your antibody. Maybe you should try and get an antibody from a different supplier Try the exact same methodology that you've done. So I did that. Same thing happens again. The antibody doesn't work. So then I'm like, okay, it must be me. But actually what you've done is you've bought. So manufacturer or suppliers can buy, two suppliers can buy an antibody from one manufacturer, rebrand as their own. You buy it as a researcher thinking that's two different antibodies, but actually you've bought the same thing. You've done the same experiment. You've wasted your time and your money.
Yeah.
And if there was
exactly
an RRID, you would have known that actually what you were using was the same thing.
And if the RRID will point to that if that information is available. Sometimes it is not available, so it doesn't always get you where you want to go. And I know that for Protein Tech, you guys have been doing a lot of work in epitope mapping. So the way that we had thought about this particular problem is make sure when you're doing two different antibodies to at least make certain that the epitope that the antibody purportedly hits is slightly different.
Yeah.
Because if you have that piece of information, you might be able to get the two different antibodies as opposed to the same antibody.
Yeah. Or even two different antibodies that don't sterically hinder. Make sure that they're at least far enough apart that they're not going to clash.
Right. Exactly. Yeah. So, and this is where the epitope piece is extraordinarily important. So if you've got a genetic model and genetic knockout, do that. Use that for antibody validation. But if you don't have that, you might use two antibodies to do the same exact thing, but you need to make sure that they are actually independent antibodies. And so, way back when we started working with some of the manufacturers, some of the smaller companies, and we said, okay, look, we're going to mark you as the manufacturers of these products. That way, at least people can say, okay, I know where this came from. I know who created it. But then even some of those companies were using what's called OEM to import different antibodies. And then it became a little bit less clear.
Yeah.
What was the what was the source of that particular product? And it's great. Many companies, including Protein Tech, are now shifting where they really don't import a lot of antibodies from others. They have their own lines. They create their own antibodies in-house, which makes some of this, it alleviates this problem.
Yeah.
The problem is there, but it starts to alleviate the problem. But, it can't alleviate it fully because I have to say, antibodies, I don't know about your hands in your lab, but the first RRID that we had actually gotten inside of a paper. So I looked up everything about this antibody because I thought, oh, this is so great. We have one. We have the first one. It's so great. It's that we've got the first one. And so I looked it up. It was an antibody that was labeled as Chemicon.
Right.
And by the time that's published in 2014, Chemicon had been out of business for many years.
Okay.
It was actually bought by another company that's called Millipore. That then became EMD, then it became EMD Millipore, then it became EMD Sigma Millipore, then I think it went back to Millipore line within Sigma. Then, and actually right now it's part of Merck.
Right.
So what antibody was that again? But again, it was an antibody that was bought so many years ago. It got stuck in a lab freezer. And people said, oh, that's the one that works. Cool. We'll keep it.
Yeah.
But by that time, the company had been out of business for a long time. So the question is, well, which antibody do you actually have? Because a lot of the products that are out there in labs, they were bought 10 years ago. They were bought 15 years ago. As long as their deep freeze is not compromised, they're going to keep using that one that worked.
Yeah, exactly. Exactly.
So you can't really clean up all of the labs and say, oh, you need to rebuy
start again.
all of your stuff. No, those are already there. They're already, and they're going to be there.
Yeah. So whilst we're on the 5 pillars, we've mentioned genetic knockdown, so the genetic strategies and independent antibodies. I think there's orthogonal strategies as well, so using Western blot and then another or antibody independent strategy, so like mass spectrometry. Trying to think, was it tagged overexpression of the protein?
Yeah.
And then final one, testing myself here, immunoprecipitation and mass spectrometry of that as well. So
there's a lot of mass spec going on, and that's another technique that's really gone. It was a technique that was used very often, I think, in the 90s, in the 80s and 90s, and then it sort of died, and it has just blossomed again because the technology had moved forward to a point where it could now check large proteins like antibodies. So it's been it's kind of found a new life. A lot of science does tend to do that. So the machinery really does drive the technology forward, and the understanding of things like antibodies forward.
So I guess the kind of moral of the antibody validation story is that if a researcher is looking to get an antibody, they need to make sure that manufacturer has at least validated in genetic knockout as a gold standard, but if not, one of the others.
At least one of the other pillars, yeah. And again, you can't validate in every circumstance as a vendor. You just can't, because there are people who are going to be using mouse antibody for fish, and that may be the only antibody that is available for that protein target. And maybe that protein target is exactly the same in fish as it is in mice. But I don't know the answer to that for every protein. There are 20,000 of them, so we can't possibly know which one is which, and we can't possibly know which ones are missing for the species that somebody was working on.
I think that kind of lends us to closing the gap between industry and academia, I guess, and working with researchers. So, for example, I know at Protein Tech, to use your example now, if we validated in mouse, but a particular researcher is working with zebrafish, which so many people are working with zebrafish at the moment, it's fantastic, but we might not have the facility to validate that. But we work with these customers, we say, try it, see, and please share your data. Let's be open access here and help, because obviously the more technology is coming out, the more even species that are being discovered still. Not everybody's going to have reagents for those, but if everybody in the world can actually just share what they have, that would be so nice.
Yeah,
it'd be so nice.
It would really be so nice. And if they put an RRID into their paper, then we could actually pull that paper back. Oh yes, it is exactly this antibody that is being used in this paper. Then you can take and you can pull some of that validation data out of that particular paper, so you can also add it to the manufacturer site. Hey, here's an external validation of this antibody in a system that we just don't have the capacity to test.
Yeah.
So what that does is it allows the manufacturers to have an additional depth of validation that they're not necessarily going to do. But of course, the manufacturer can't be off the hook for basic validation.
Yeah, of course.
And if they can say, hey, here's the basic validation. Here are the circumstances. Again, here's that method section for how this was validated, which a lot of companies do have. Some companies don't. And you ask, how did you do this? What species is this in? What sex is this in?
Yeah.
Because some proteins are not expressed in both males and females, right? So, the reproducibility crisis and the ability to have validation data for these reagents, it kind of goes both ways.
100%.
Yes, the scientists are definitely not blameless here, right? We've done a terrible job in many cases. Figuring out or validating our reagents. We've done a terrible job even identifying the reagents. In the past, we're starting to do better, as I've said, and we've got some lovely evidence for that, which is great. But and a lot of the companies have also done a terrible job in putting that data in front of people. They might show a Western blot, but then they might not have the depth of evidence for what was done there in that Western blot. How was that performed? Which cell line was it on? Or was it a cell line? Was it homogenized tissue? Was it, what was it?
Yeah.
I see some bands. Okay, cool. What does that mean?
Yeah.
Right? You might not even have the latter to show what is the size of the protein. You might not have that in the same Western.
Yeah.
So, there is evidence there, and there's kind of culpability on all sides, but there are also good people trying to do the best that they can with what they have and trying to improve the situation.
Yeah.
And this is where that, again, that ship, that very large ship, needs to right itself.
Yeah.
And I think that, when we talk about reproducibility crisis, when we talk about these things, yes, it exists, right? We have some problems with reproducibility, but we also have been alerted to them. And it takes a while to get so many millions of people to start doing a slightly different thing.
Just to be aware of it, I think.
Yeah.
And aware of that it's not their problem, I guess. Yeah. Like just young researchers going into the lab, that's something that you need to think about, good practice, and knowing what reagents you're using. Going back to the rogue antibody that was found in the back of the freezer 10 years ago, that one person didn't know. It was the one that worked, but where can I get it from? At the moment, there's a big influx of recombinant antibodies and sequence definition of antibodies. Do you think that moving very slowly but potentially exponentially in the direction of sequence-defined reagents is going to help in the sense of people actually know what they're working with?
Absolutely.
Yeah.
I mean, this is something that we really have to come to grips with. A lot of people who really love their antibodies just are unwilling to change. But again, we have newer researchers, we have younger researchers that are coming in, and they're going to be the new ones. So they're going to potentially have to buy new antibodies. And hopefully those are the recombinant antibodies. We know so many more things about those specific reagents. I mean, a lot of the validation problems was, and if we take this concept of the chemicon, Upstate to Chemicon to Millipore to every other company known to man, antibody, it's a single reagent. It was a monoclonal antibody, and monoclonal antibodies are essentially a cell line that produces the reagent.
Yeah.
Not a known message, but a cell line. And cell lines are alive. They're living things. They are something, because it's a living thing, it will mutate.
Yeah, it's got susceptibility to genetic drift.
Exactly. So you start to get genetic drift. And if you don't treat them very well, which you might not treat them very well if you're going to lose your job next week.
Yep.
So if you're going to lose your job next week, you might not treat those cell lines extraordinarily well. And then whatever is being produced out of those cell lines is going to be bad. Mm-hmm. And in fact, this did happen to this particular antibody.
Okay.
And the thing that happened to this antibody is that it was first licensed out of, so there was one antibody that was licensed out of an individual lab. And it was licensed out by a company called Boehringer.
Yep.
And Boehringer then became Roche. Roche sold off its entire line of antibodies. They licensed a bunch of stuff back in it later, but that's neither here nor there. But this particular antibody had gone through one of these, dreadful situations where it was moving from one company to another. And at one point, the author of the original antibody paper, which, this reagent he knew and loved, he bought it and he said, this doesn't work anymore.
Wow.
And so he had to write a paper to let people know that this antibody doesn't work anymore. And it didn't work anymore, probably because someone was losing their job. And I don't know what they did with the particular culture, but it was bad.
Right.
Maybe it was negligence. Maybe it was intentional. We'll never know. but the product didn't work anymore. And they had to go back and rederive it from an earlier stock. And thank goodness that the antibody was rederived and was able to work again. But this is a vulnerability. When you have the sequence itself, you can create that in a different kind of cell. You don't have to have a cell that is susceptible to genetic drift. And you can more reliably create that product. I'm certain there will be more things that will be able to be done, as we're using AI to actually create a lot of these proteins.
Yeah.
And we're starting to understand much more about how to do these things.
Even just the backbone of the antibody, knowing the backbone sequence of that antibody, you can engineer it indefinitely. Essentially, you can do so many things that you can put in conjugation sites that are site specific. You can take away Fc binding.
Mm-hmm.
You can eliminate things that researchers might not even be aware of. You can, for example, at Protein Tech, just to have a little plug there, we've got our FC0 backbone, which it's recombinantly defined. We've mutated the Fc binding portion of the antibody, so you no longer have to block in your flow cytometry pipelines. The benefits of that alone are exponential for things like therapeutic pipelines. If you're, making a therapeutic, if you're wanting to build ADCs, bispecifics, it's just, it will snowball.
The fun thing about the therapeutics, antibodies, when I started this, really had nothing to do with therapeutics whatsoever.
Right.
And in the last 10 years, we've gone to a majority of the blockbuster drugs being antibody-based, which is huge, huge, huge difference in terms of where we were versus where we are today. So all of our blockbuster drugs, all the MAB ending things are actually antibodies. But some of the problems that we just described with cell lines not being true are going to be exactly the same problems that these big drug companies are now facing, because they're going to get genetic drift in those cell line populations that are creating your blockbuster drugs.
Yeah.
And they do have to go through a lot of generations of those cell lines, because they're having to provide a product to an entire world.
Whereas recombinants, very scalable. Whereas
recombinants are where a lot of the newer companies, or a lot of the new drugs are actually moving. But which those are much more scalable. They're able to be manufactured in cells that are not necessarily cell lines. They're not going to genetically drift. So they're a far better product.
They also can rise against any sort of geopolitical thing, because you can send that sequence in an email. It doesn't have to go through any sort of border control or anything like that. So they're safe. They're safe from anything that we might potentially come up against in terms of scientific supply and all that kind of stuff.
Oh yeah.
So I guess in the world we've seen a few examples. You've brought up a few examples of where having a persistent identifier of a reagent is really kind of useful. Have you got any other examples of where having specific reagent identifier is really useful?
Oh yeah, there's a lot of examples, but one of the things that we did is we actually looked at cell lines. And as we've just been describing, cell lines, when they are used by people, or even taken into a manufacturing facility, they can drift, right? Drift happens, genetic drift happens. And some of the things that have been documented are HeLa cells, in The Immortal Life of Henrietta Lacks.
Great book,
great film. But, one of the things that was discussed there, which probably many of you missed if you aren't in the antibody space and in the cell line space, one of the things that was brought up is that, HeLa just takes over everything. So the way that I usually talk about this is, if you have a HeLa cell and an undergraduate in a lab, what happens is they'll sneeze at some point and everything will become HeLa. So it's bad. Don't do that. But, we do train undergrads. So we have to be aware that HeLa cells can kind of take over other cell line colonies. But there are known problems before they get to your lab that some of these cell lines are known to be contaminated or problematic in some other way. One of the cell lines that's really commonly used, and this is funny because I was just talking to a colleague of mine this morning, and we were talking about the HepG2 cell. HepG2 is a really commonly used cell line, but it has a note on it. And what's the note? Well, turns out that the HepG2 cell, when it was taken out of the patient initially, it was diagnosed as a hepatocellular carcinoma.
Right.
But it's actually a blastoma. So it's a different cancer than it was originally thought to be.
Was that a misdiagnosis or is it evolved?
It is a misdiagnosis.
Okay.
And we know in the world of medicine that we get misdiagnosed.
Yeah.
Right. And so it kind of stands to reason that if you're taking a sample out of a patient, some of those samples are misdiagnosed.
Yeah.
So this is one of those cases where there is a misdiagnosis that was originally written down as part of the metadata for that sample. And that had propagated. So everyone who uses that cell line might be using it for the wrong disease.
Wow.
Which is kind of crazy. So my colleague, who is extremely aware of antibody validation, cell line validation problems, he knows all about the International Committee for Cell Line Authentication. He's keenly aware, and yet he used that particular cell line. And he had knocked out some genes out of it and had given it to his colleague to use for some antibody experiments. So now you have a person who is extremely aware of all of the antibody validation issues and all of the other validation issues, who wasn't aware of this particular note. And so what we had seen when somebody is writing their paper, when they are starting to create that manuscript, which is going to be ensconced in the world biomedical literature, right? And they are provided that bit of information, hey, you might want to take a look at this particular cell line because it was originally misdiagnosed or some other issue. Then what you actually get is you get about a 66% decrease in the use of those particular cell lines because, again, people they don't want their reputation to be based on faulty reagents.
Yeah.
They want to put their best foot forward. And so what they will do, even at the kind of latest stage of publishing, they tend to pull those things back. And they tend to try and fix the literature before, again, their reputation is staked on this kind of a faulty reagent. Mm-hmm. So what happens is we think we have an intervention at least one stage by having the RRID system actually have the piece of information that is so important to that validation of those particular reagents. And we have evidence that people see those and they act accordingly. Mm. So this is just one of those cool things that we were able to see, to detect. And if people are publishing with this kind of a reagent and they see a particular problem with that reagent, they can still pull that information back.
Yeah.
They don't have to publish it. They can, or they can rewrite their paper to, ensure that that is not adding to the noise, right, of the scientific literature. So the RRID system, because it's got these notes, And we're trying to put these notes out across all resource types and reagent types. So we have notes about antibodies that are validated. We have notes about antibodies that are not working correctly.
Okay.
As far as some scientist out there found they're not working correctly. We have notes about cell lines that have been either contaminated at the source or they've been misdiagnosed in this sense. We even have notes about, the most common mouse that is used is the C57 Black 6J.
Okay.
So this is the Black 6 mouse. Everybody uses it. They all think it's a control. But there's one scientist who decided to look at the prevalence of the GABA A receptor, which is the main inhibitory receptor in the brain, for these mice. And she looked at the N strain, which is the other C57 Black 6. It's the that's not the one that comes from Jackson Labs. The one from Jackson Labs is the J strain for Jax. We're not very good at naming these things, but in any case, so if you look at those two strains, which have the same progenitor, one of them has about 25% prevalence of GABA A receptors compared to the other. And that J strain, which everybody uses as a control, is actually epileptogenic.
Wow.
Because of that low prevalence of the GABA receptor.
Okay.
So if you're using that mouse as a control,
don't be studying epilepsy.
Don't be studying epilepsy. Study something else, but don't study epilepsy because that's not a good control for you.
Yeah.
So we have a note, again, showing that, okay, if you're going to be studying this mouse and you think it's a control, good. I hope you're not studying epilepsy.
Yeah.
So, but the same is true for, software tools. We had talked before this about software tools and people not getting proper credit. Well, there's also similar things going on with software tools. There are software tools that were created out of a laboratory. Those software tools had a bug in them. Remarkably enough, we all have seen bugs in various bits of code. Some of those get out before the thing is published or before the thing is sold, but many of them aren't. There are all these patches and things that we have to do to clean up these bugs. Well, what if that bug actually changes a Fourier transform?
Yeah.
And we have some of those examples of code that, for example, doesn't work the same on a Mac versus a PC.
That simple.
That simple.
Oh my gosh.
Yeah. And then the question is, all right, who used that code? What were the results of the study? Yeah. And could this bug have actually made an impact on the results and on the discussion on what came out of this particular study? And again, because we haven't had good ways to identify the resources, we can't figure out necessarily what the impact of those kind of bugs have been or has been in the scientific literature. So first and foremost, please identify all of your research resources, including antibodies and especially antibodies. Mm-hmm. But if there are problems that have been discovered and you may not have discovered them yet, like my colleague who didn't know that Hep G2 cells were actually originally misdiagnosed. And as we were talking, he was like kind of concerned about the fact that maybe his experiments are wrong. Yeah. And I was like, and you are in the know.
I was going to say,
know everything about this field, and you didn't know that one piece of information. And it is difficult to know what everybody else said because we don't publish about these things specifically, usually.
So you started the RRID initiative within the last decade? Mm-hmm. Oh, gosh, time is going so fast.
11 years.
11 years, yeah. What has been... Obviously, you mentioned there that somebody who should be in the know didn't know everything, but what has been the sort of layman uptake of this, or not just any sort of researcher uptake of this, and how have you tried to implement it into the scientific community?
So the uptake has been wonderful. We started out with 25 journals in 2014. By the end of the first year, we did have actually 25 journals when we had about 100 papers in that first year. And you might say, oh, 100 papers, that's great. We did write a lovely paper about that, but that was fine. But the point was 100 papers out of how many?
Yeah.
So each year we have, about 1 million papers being published, and that rate is growing. So in 2014, we had 100 out of that. In 2015, we had about 300 or 400. Then we went to several thousand. And by 2022, we had 500,000 RRIDs inside of the literature. And we're really proud of that. We're so excited. I wrote a paper about it. And I was just rah. It's like, hey, it took us, 9 years to get to 500,000. And then within 2 years, we were at 1 million.
Wow.
Which is great. So there is an exponential uptake, which is great. And it's because people don't have to be convinced that, yes, they need to write down which reagent they're using.
Yeah.
It's something that is just part of science. But because of the way that publishing evolved at a time where there weren't that many reagents out there, people would put into their manuscripts, the company name, the target, and where the company was located, which is so useless as a piece of information when you're going to an online catalog. I remember when I was a grad student and I was putting in this information and it was some multinational, right? Sigma. I was like, I'm really concerned. Is this information wrong? Because I don't know. Is it their headquarters? Is it where we bought the reagent from? Like, what am I supposed to do
PO
there?
Box.
But it's like, oh, there's some random PO box in Cayman Islands or whatever. And who cares, right?
Yeah.
The pertinent piece of information there is the catalog number and the lot number. Yeah. It's not where the company is headquartered. I mean, that doesn't help you anymore. Yeah. That used to help you when you had to call the right circuit board. Call up Betty and say, please connect me to, Maryland somewhere. I mean, Betty's gone. She's dead. I'm sorry. She's been dead for many years. The phone operators are not going to help us get our reagents anymore. We now have online catalogs, this thing called the internet.
Yep.
Yeah.
So that old thing.
You can buy toilet paper, you can buy ice cream now, big thing. And you can buy antibodies.
Amazing.
And all you need is the catalog number.
I suppose it goes back to kind of good research practice and how people are taught initially. Because I know when I was, going back to me again, when I was in the lab, the only time that I came to thinking, right, I need to tell people where I got this antibody was from, was when I came to publish, which is, 3-4 years into my career. And I go, can I remember, did I write that note down? Did I even think that it was important? Nobody told me that. Like, should I have been thinking that myself? But obviously good research practice should include where exactly all of the different recipes for that meal that you prepped were from. And you should know that right from the outset. I think from previous conversations, we've talked about bioarchive and an AI tool that you've generated to identify.
Oh, yeah. So even though we told NIH in 2010 that we really couldn't build an AI that would actually do what we were asked to do, we ended up building that at a later stage. But our goal was to, again, intervene with the author to get them to clean up their paper. Yeah. And so we built a tool called SciScore, which intervenes with authors during the publication process, helps them get their RRIDs in. It helps them remember other things that they may have, may have slipped their minds. So it's kind of a compliance tool for rigor and reproducibility. And we took part of that tool and opened it to the public by processing all bioRxiv papers. So we now have a little bit side to each of these bioRxiv papers. And we put everything up on Blue Sky so that it's posted about. And I think we have, 20,000 things, papers, preprints that have been processed at this time. So we're really excited about that. But the whole goal is to make this process, one, to make it easier for people so that they would think about, like you just said, you had to think about that because it was 3 years ago, and did that piece of information exist somewhere? Can I go back to those records? So we give them some of these things. We give them the guesses if they put in the amount of information in that they can, where there are the, essentially if the AI feels comfortable enough that there's enough information there, then it'll bring back an RRID for them.
Okay.
If not, then we want to flag those places in the paper for the author to say, hey, you might want to think about this because we're missing a piece of key information.
Yeah.
And that's the bit that I really think that AI is going to be good at, because you're not going to have individual reviewers, individual people going through each of these catalog numbers, right? And saying, oh, did they put this one in? Is this one accurate? Did they put this one in? And if they have a list of 100, there's no reviewer on earth that is going to actually go through each of those. But that's where you have AI tools. AI tools are not going to care whether you've put in 1 or 500 of these things in. The tools can just be there, and they can check each and every single one of these and say, this one doesn't match up with something. You've got a problem. And then the attention of the author can be focused on just that place where they may have made a typo, a mistake, an error of some sort. And then that error doesn't get propagated, doesn't get published.
I was going to say, is this sort of like a predictor of reproducibility then, like from the paper?
Yeah, we are actually working on some predictors of reproducibility. Actually, we're working on predictors of also retraction.
Oh, really?
Turns out that if, we have to... Well, we
can hear scientists quaking in their boots now.
Well, I mean, it seems to be, and I need to find whether this is going to hold true in larger data sets. But from some preliminary data, it certainly appears that if people are putting in a lot of RRIDs and they're accurate into their papers, those papers are less likely to be retracted, right. Which is not something that would be revolutionary, right? If somebody cares more about their method section, chances are that they're also going to care more about the rest of that paper.
Yeah.
And so it's kind of a mark of quality, in a sense.
So this size score is a tool for essentially a little bit like a plagiarism check was what I was looking for. The checks that I used to at uni, like you'd pop your document through and it'd check for plagiarism. This is like basically another one of those where it's
another one of those.
You're putting this document through, you've already written it. It's another step. It's going to help you hopefully with the reviewer process, which is always welcomed.
Of course.
It pops out a score of, which
things are missing, which things aren't labeled correctly. It would get people to think about getting their sort of house in order, in a sense. And if there's something missing, they need to go track that down. But they might not be aware that it's missing. I mean, if you have a big section of text, so if you have a table and there's some information missing, it's really clear, right? It's like, oh, this one has all of the fields filled. And the next line is missing these 2 fields, and they're blank, they're white. right? And you're like, oh, okay, there's missing data here.
Yeah.
That's easy to tell.
Yeah.
Which is the real power of those cell press tables.
And this is available to researchers when they put it onto the bioarchive preprint.
Yeah, absolutely.
Is it going to be something that is rolled out to other journals, like the full publication processes as well, or is it a case of just so it's growing?
Yeah, actually, so we're working with quite a few publishers now using that AI tool before the paper is published, and it's either transparent to the authors, where the author is actually running the tool until they kind of hit the score that they need to hit for the journal.
Yeah.
For the the bits of compliance that they need to do with journal policies, or it's being run by the journal themselves.
Right.
And the journal sees whether the author has hit that score or not. So it's kind of fun.
From which side?
Well, maybe more from my side than from the author's side. But the point is that the journal is going to get back to those authors, and they're going to ask the author to fix things before those things make it into the record.
Yeah.
Right. I mean, the journals, again, like we had talked before, there are people who care about rigor and reproducibility. They care about science. They're really deeply passionate about fixing the reproducibility crisis. And those people exist at antibody companies, in academia, in journals, in all of these different places. They care about fixing this because this is not just an economic engine. This is not just, the ivory tower. This is the engine that has given us a whole lot of all of the new medicines. It's giving us a lot of the technology boosts and shifts in the last decades. This is the thing that drives us forward.
It matters.
It matters.
Yeah.
Right? It matters. So we want to do the best we can with whatever we've got. And a lot of people do want to do that. And so, I don't think any of these things are at odds. I mean, yes, you get the individuals that are bad actors. And you get them everywhere. We won't name any names here. But most people want to do the right thing. And if you point something out, hey, you've forgotten this, there's a bug here, there is, there's an issue with this reagent, they want to fix it.
Yeah.
They don't want their name to be associated with that.
What do you think the kind of next steps are for increasing the awareness of this is how you should actually be writing your papers and this is actually how you should be carrying out your research? So
we've looked at that as and we have a preprint out on this exact issue. Hopefully it'll be published soon. Please, plus. let's get this through. But we've looked at that issue, and so we know that there are all the different stakeholders, right? There are individuals who just do this as a matter of how they publish, which is great, but that's a small number of people. Those are people who have heard of RRIDs, who like RRIDs, and they're always going to use RRIDs no matter where they publish. Awesome. There are the journals, and the journals say, do these things, be compliant, and, publish with RRIDs. And some of them even stand for the fact that without these RRIDs, you're not going to publish your paper. Most journals don't do that. If you miss one or two, they'll let you publish. But they're going to encourage the authors to do that publication. And then there are the companies themselves, or the reagent providers, the resource providers themselves, who say, if you're going to use this, here's how you cite this particular product. Here's how you cite this particular reagent or mouse or whatever software tool. And those are those are kind of the third stakeholder. And we've been able to track down what percentage of RRIDs in the literature come from kind of individuals versus journals versus those manufacturers, those reagent and resource providers. And journals drive about 10%.
Okay.
And the about 20 to 30% of the antibody, at least the antibody and organism data comes in because the projects, the manufacturers, the companies have on their websites and have in their material data sheets those RRIDs. Here's how you cite this thing. So we're getting compliance now. In some cases, over 50% of the papers are compliant, which is amazing.
Yeah, really good.
It's really good. I mean, 11 years. It seems like such a long time, but
in the scientific world, that's a short amount of time.
Sure. Yeah, again, to turn a ship that size, it takes time.
Yeah.
And in that short of amount of time, we've gotten above 50% in many cases. And to get to that 50%, a lot of it is driven by the manufacturers.
So kind of open access, making things really accessible for the researchers. So you mentioned there, obviously, the publishers and the manufacturers. Is there anything that we can do for the fundamental researchers, that kind of early career researchers as a staple? Obviously, these people are coming through. Like, how do we get that into their brains?
I love doing podcasts
There you go.
and other educational materials are out there. I mean, RRIDs, add them to your paper, please. I run a tremendous number of workshops. We work with early career researchers. We work with mid-career researchers. We try to do what we can in our lab to get the word out, right? And so we truly appreciate having any kind of a stage to be able to reach more of those researchers, because at the end of the day, they're going to have to be the ones who look up their reagents. They're going to have to go back to their lab notebooks. They're going to have to go back to their inventory records, and they're going to have to find that information.
You mentioned lab notebooks.
Yeah.
More and more people are moving to online notebooks now. Is there hopefully going to be some sort of plug-in that we can just drop down your RRID?
Hoping and praying. So we've been trying to work with some of the laboratory notebook systems to be able to actually pull that together and add RRIDs at earlier stages before you do the actual experiment. And there is one open source notebook project. The others have not yet come along. We've been trying to talk to them for a long time about, hey, don't make people write all of this metadata. Some of it is going to be wrong. Instead, pull it from us, because if ours is wrong, we'll fix it, because we have all of the manufacturers on board. Not all of the manufacturers, but when the vendors are on board, we can at least fix the data if there is a problem. But in your own lab notebook, why would you have to put all of that metadata in
when there could just be a little code?
There could just be a little code that would just pull the data from us.
Yeah.
And it's free and it's open, and yes, they can do it. And no, they don't have to pay for it. They just need to pull all of the metadata. So, so for the early career researchers or mid-career researchers, tell your lab notebook provider that you need RRIDs and you need them to hook up to the RRID services in order to pull the metadata so that you don't have to fill it in. Then maybe they'll listen.
Yeah, we need everybody involved. Rally together, everybody, to bring those RRIDs. So I'm interested, if we go back to when you were at the lab bench, if you think back to when that was, is there anything that you would do differently then, given what you know now about persistent identifiers?
Yeah, I would certainly put more persistent identifiers into my papers. I mean, I look back at some of my papers from way back when. I think they were dinosaur papers. But, as I said, the kinds of things that I put in there were, Sigma Aldrich, where is that company headquartered?
Yeah.
All of those bits of information I didn't realize were not useful. They were leftover. They were how I was taught to write.
Mm-hmm.
And they weren't good practices necessarily. But they were the practices that everybody was used to. I mean, I only used male rats because I was told by someone that female rats had hormonal problems. And I'm like,
excuse the data.
Yeah. Okay, sure.
Could be useful.
Could be useful, though. And it turns out that when you actually try to translate, findings that were found in male rats to humans, the drugs that we have working in, this one strain of rat actually doesn't translate well, especially to women.
Yep.
And remarkably enough, people of African origin.
There you go. I was actually going to ask, are these RRIDs, if we use this system, is it going to increase, diversity and equality and equity of representation?
I would love to expound upon that as much as possible. But in fact, RRIDs themselves may or may not help diversity and inclusion. But one of the things that we do need to do as a scientific enterprise, we do need to increase the substrate that we are studying. We can't be using just one strain of animals. We actually need to increase the diversity, the genetic diversity of those animals in order to determine whether a particular drug, a particular treatment would be better, would be more generalizable to larger diversities of populations that have to larger diversity.
Yeah. I mean, even not just on the animal level, in the sort of phase 1 trials or something like that, first in human trials at the moment are still all, done on a mixture of sexes. Mm-hmm. And then the data is extrapolated for both. Whereas if you could just actually look at drugs separately and attribute their utility specifically to that particular subgroup, surely that just makes sense that it's going to be more efficient.
It does. But, one of the reasons why a lot of drugs fail on again, women and people of African descent is because there's a much greater level of genetic diversity in the people of Africa, because the rest of us came out in sort of a small, in a much more of a single migration.
Okay.
And so you are much more related to Asians and even Native Americans than you are to the current African populations.
How interesting.
Which is one of those weird things about going back to, a field of science that, is seemingly so outside of cell cultures and drugs and what have you, but it all impacts. And the impact of the migration of humans is one of the things that actually drives how drugs work right now, which is odd.
But it makes sense when you put it like that. But it makes
sense when you think about it, because if there is more allelic variation in a particular place where those drugs are going to be having their impact, then, and if you don't have that group represented well in your clinical trials, then once those clinical trials get bigger and they start to include those people, now you have the problem of, okay, now it's not working or it's having a toxic effect on this population. Mm. So again, inclusion in this sense, we want to include people of all races, all backgrounds, so that, or at least we need to know which people is this particular drug going to be able to be curative for?
Yeah.
Is it going to be toxic for those people? And, we need to understand that. And we do understand that when we move to the larger trials in general. But earlier in animal research, you should be able to model some of those population diversity issues. And we are often not doing that. So, my other plug in this is, name your reagents, but also make sure that when you're using animals, you can use not just a single strain.
Yeah.
Try to use multiple strains, because then you can understand how generalizable a particular effect might be.
Yeah.
And maybe if you use multiple strains, your effect washes out to nothing. And that's fine. Now you have prevented a clinical trial that would fail later with humans.
I suppose the use of RRIDs, whilst it might make it worse in a way that people have their favorite RRID or something like that. What it might do is, if everything that you use in the lab has a persistent identifier, you can then down the line identify trends in what is being used, what needs to be used, and what holes can we fill in the reagent space in order to generate that kind of wider impact down the line.
Well, one thing that just kind of came to me, if you think about RRIDs, if you are using them for each one of your strains for animals or any of the other reagents, but if you think about this in terms of strains, what you would actually be able to do is say, okay, how many different animals was this particular drug tested on before you get to the human clinical trials? And if the answer is 1, well, maybe do it in a couple of different strains before you get to the humans.
Yeah.
Because most likely you're not seeing something in terms of the richness of the allelic variants that you might need to account for before you hit human clinical trials. So, I have never thought about that as a signal for quality, but I think it's something that we might want to look at.
Definitely. No, well, RID Amy Turner.
We'll credit you.
I suppose that leads me on to one of my final questions in the time that we have. Where do you see the next sort of 5-10 years of your career, but also the wider impact of RRIDs taking you?
That's always a really hard question. I mean, I would not have put us at where we are now 5-10 years ago, but it's certainly things are moving and improving. We are getting to now have a new standard for how do you talk about antibodies, where at least people are putting in the catalog numbers for nearly all of the reagents. I mean, we're still seeing some papers that don't have that, but it's now rare, whereas it used to be the norm.
Right.
So I think that we would go that next step in the next 5-10 years to where it's now completely normal to look up your RIDs, keep them in your lab notebook, whether electronic or otherwise. That those persistent identifiers actually help pull the metadata that's the correct metadata, as opposed to having to enter it in every time. I definitely see some signals that might be derived out of the RRID literature now that it's getting to be more substantial in size. And we're just going to start to understand how to use that data now that it is not a mere project that's small and nice. But it's starting to become much more mainstream.
It's kind of fundamental.
It's starting to become fundamental. So I think we will see more uses of that data now that it is substantially larger.
And accessible.
And very accessible.
Yeah. Okay, final question. You've had a wonderful career. I would dream to have this kind of career from kind of research all the way to literally influencing global research. Has there been kind of one moment in your career that you've kind of can highlight, that you can pinpoint and go, this is why I do what I do?
Yeah. So I was talking to a colleague who was, an antibody manufacturer, actually, or working with an antibody manufacturer. And he said, yeah, the reason I quit science was because of an antibody not working. And I said, really? And he said, yeah. I wasted 2 years of my life trying to get a PhD, and the antibody wouldn't work, and the antibody wouldn't work. Finally, after about 2 years, we figured out that the antibody batch sat on a tarmac for 3 days and became useless. That batch of antibody destroyed that career. And there were others like this that again, people who pull me aside and say, oh, I've got this horror story. I've got this horror story. I really think that was a thing that drove me to help those people, because they could have made very different contributions without having to fight with these reagents all the time. I think we need to stop fighting with each other so much. I mean, there's going to be differences of opinion, and that's all great. And we'll need to fight in the right way, right? We shouldn't be fighting with reagents. We should be fighting with ideas.
I love that. Thank you so much for this conversation. It's been a wonderful few hours, and I really hope that the listeners of this podcast will start using RRIDs and just have a think about how your research practice is really important for the continuation of both your projects and the projects of wider people. So yeah, thank you so much for your contributions. It's been wonderful.
Thank you so much for inviting me. I really appreciate this.